Hi, I'm James Graham, CEO and Managing Director of Recce Pharmaceuticals Limited. Today, we take a deep dive across Recce's anti-infective portfolio, brought to you by experts across their respective anti-infective fields. Beginning will be Dr. Alan Dunton, with insights as to the phase 1 intravascular study completed at CMAX clinical trial facility, particularly focused upon interim PK data, achieving primary endpoints, and the next steps of a phase 1b, 2a study, as well as a new phase 2 UTI efficacy study in humans. Following will be Dr. Mark Sharp of Linnaeus Biosciences. Dr. Sharp is a world-leading mechanism of action expert in antibiotics who has examined Recce anti-infectives to investigate its potential against the full suite of escape pathogens, its biofilm capabilities, its abilities against both growing and stationary phase bacteria, plus its ability to work against World Health priority pathogens. Dr. Philip Sutton, former Murdoch Research Institute, will expand upon our preclinical datasets, Bacterial Sinusitis, H. pylori, and M. abscessus. Following will be Guillaume, Managing Director of LifeSci Advisors, who will look to the commercial opportunity that this suite of anti-infective pathogens and new classes of antibiotics may face. There is a recent deal done by GlaxoSmithKline in the UTI space of around 100 million Australian dollars. There is expanding market opportunities and market interest for new classes of antibiotics. And we will conclude with Michelle Delizia, Executive Director and Chief Scientific Officer, looking to the full therapeutic roadmap of Recce 327, the significant and unmet medical needs Recce's anti-infective portfolio seeks to participate in, and the multiple clinical stage therapeutic indications this company now has. We thank you for your time and look forward to introducing Dr. Alan Dunton.
Hello, my name is Alan Dunton. I'm a non-executive director at Recce Pharmaceutical, and I've had the pleasure of working with the team at Recce for a little over 2 years now. I joined as a consultant early on and later became a director. My background is in medicine. I am a physician in adult medicine, but my specialty was in clinical pharmacology, which is the action of drugs in humans. And as such, I pursued a career in drug development, which was very satisfying, a 40-year career. And now it's a pleasure to be helping a young company just like Recce. I used to be the president and managing director of Janssen Research for Johnson & Johnson. You probably have heard of them. And before becoming the president of Janssen, I was the global vice president of R&D for the R.W. Johnson Pharmaceutical Research Institute, which was another arm of drug development at J&J. What I'd like to do this morning is take you through drug development as seen through the eyes of Recce. I've followed this prescription many times, and it's been successful very frequently. As you know, we're developing an antibiotic. It's called Recce 327. And the way an antibiotic is developed is screening occurs not in animal models, but in cellular systems. And you can actually evaluate whether your drug is efficacious in killing various bacteria or a virus or fungi or capsule species. And then you apply them to animal models to check on the data when you see a strong signal. So we saw a very strong signal against the E. coli bacteria, which is the offending agent for most forms of infections in the urinary tract. And if I can take you to the next slide, we did this study in rats where we infected them with E. coli, actually in through the bladder, and the bladder became infected, as did the kidneys and the ureters. And what this slide is showing on the left, when it says efficacy in bladders, it's actually the amount of bacteria that's present in that bladder tissue. So the bladders are removed after the rats are dosed at a certain period of time, beyond 24 hours. And you can see the first gray bar is the vehicle control. They were infected animals. They had a placebo, so they really didn't get treatment. And in the blue bars was a lower dose of Recce 327, 50 milligrams per kilogram. And you can see a rather significant, greater than 1 log reduction in the amount of bacteria present in that bladder tissue. And when animals had been dosed with 500 milligrams per kilogram over that 24-hour period, the decrease was even more dramatic, down to 6.69 logs was present, so greater than a 2.5 log reduction in the amount of bacteria that were in the bladder. And then we did the same evaluation in the kidneys. We ground up the kidneys and looked at how much bacteria were present. And what you can see there at the higher dose, the 500 milligrams per kilogram, there was over a 1 log reduction actually in the kidney. This is really excellent results in this model, which I've used before. So this rang the bell, obviously, that perhaps we have a chance to be an effective antibacterial agent with this product. But obviously, what you have to do then is to go into clinical trials, which we did this year. We went into the phase 1 trials, which look at the safety of your product, as well as the pharmacokinetics, which means how much drug is present in the bodily tissue or sample of fluid that you're taking. So we looked at plasma levels in the blood as well as in the kidney. I'll have to say that the drug was very well tolerated, but what we found, which was very, very pleasant for us to see, was that the primary route of elimination of 327, meaning how does it get out of the body, appears to be through the kidney, into the ureters, and down into the bladder before it's excreted. So if you look at the bottom of this slide, it shows you the concentrations on the left-hand side in human plasma of 327, and on the right-hand side in human urine. And these are comparisons within the same patient, obviously. And as you'll note, in the plasma concentration side, as we went up in dose from 50 to 6,000 milligrams given intravenously, we saw a corresponding, and I'll call it a dose correlated, meaning it went up as much as the dose went up in the plasma concentration of R327. Now, these plasma concentrations are more than enough to be treating bacteria, to effect a kill of bacteria, or in fact, potentially even virus, in the human condition or in the human system. But really was a bit of a surprise to us, because we hadn't seen this type of thing before, since we hadn't done human trials, is those concentrations in human urine. And to the right, you can actually see the ratio of the urine concentration to plasma. I'll call it on average about a 15-fold higher concentration in the urine as compared to the plasma. So if you're thinking about developing this drug for urinary tract infection, you're in the right place, because the drug is apparently cleared through the urinary tract. Now, we suspect it's being distributed throughout the body, but it clearly seems to concentrate in the urine. So this is a real opportunity for therapy, not only of UTI, but from the plasma concentrations. Could be used as a broader anti-infective treatment for other conditions, and we'll be testing those as we go forward, in addition to urinary tract infections. The next slide is the same single-dose study, which shows the concentrations in the urine, and you can see the dramatic increase as we go up in dose. In the slide on the left, the purple bars are the actual concentrations in human urine, which is, let's call it, its maximum is about 2,200 micrograms per mil, and this is a very high level. Now, what's very important, and I asked myself the question, yes, but was it tolerated in the bladder and in the kidneys? It was very well tolerated. There was no sign of any irritation or any other issue with the urine that we noted. There were no red cells in the urine, no blood, obviously, anything like that. Patients did fine with this drug. So it's not only well tolerated in the body for all the other things we looked at, but in the urine as well, in the bladder and through the urinary tract. So this is a very compelling profile for an urinary tract infection drug candidate. I've developed several products for urinary tract infection, had those indications, where that indication, and this is a very exciting result. And again, here are the plasma concentrations. And as you can see, a very nice rise as you go up in dose of the human plasma concentration. And again, as I noted earlier, these are reaching plasma concentrations which can be efficacious to engage bacteria, not only in the human vasculature, but also in other parts of the body, as we are suspecting that it's broadly distributed throughout the body. So in summary, then, we've completed the preclinical studies in UTI model in the rats, which, as they say, follow the data, and that's what we did. We saw a signal in the rats. And then again, in the phase 1 clinical trial, which we're completing, given as a single dose, R327, the drug was safe, well tolerated, and we saw very high concentrations in the urine. So another very positive signal for not only treating urinary tract infections, but as we know from the data that the E. coli is the most common bacteria which causes sepsis in humans coming in through the urinary tract system. So we have very strong data in E. coli and other bacteria. We have high concentrations in the urine, so we'll be doing phase 2 trials as well. The thing that's very important here and that's critical is we feel we have a chance to have a single dose cure using R327 with these high concentrations, plus the mechanism of action, as you've heard by other speakers, mentioned by other speakers. We have a real shot here of a single dose cure. There's a very high percent of, especially in women, who get chronic urinary tract infections. They're put on many different antibiotics, which eventually all they do is make the E. coli resistant to the new antibiotic they're being put on. Given our mechanism, we feel this is not going to happen with Recce 327. So we'll be doing single and multiple dose evaluations of the urinary tract infection, different types of UTI. And then with a positive signal there, we can move into what our ultimate goal is to have this drug in sepsis in patients who are very sick. So they'll be in an ICU type situation, and they'll be evaluated for the efficacy of 327 in urosepsis. So there's a very exciting period of time for the company moving forward. And, this is one of the best drugs I've seen in terms of a urinary tract infection candidate going forward. So, I've had fun in this career that I've had in drug development, and it's been a pleasure working with Recce, and especially with Recce 327. So I thank you for your attention, and have a good one. Take care.
Hello, I'm Mark Sharp. I am the Chief Scientific Officer at Linnaeus Bioscience. We're a company that uses some proprietary technology to study the mechanism of action of new compounds in a wide range of bacterial species. And Recce approached us a little over 2 years ago to start looking at the mechanism of action for Recce 327. And it's been a very exciting compound. It's got a unique mode of action that we've not seen in any other compound before. So I'll take you through some of the results we've seen and what that's telling us about what Recce 327 is doing. Recce 327 has a very broad spectrum of activity. So it's active against both gram-positive and gram-negative bacteria. It also affects different mycobacterial species. We've tested it against a subset of the escape pathogens, Bacillus subtilis, which is a gram-positive bacterium, and Escherichia coli, both in biofilms and also in just free culture. And it's effective against all of these different species in different conditions. I'll be going through a series of graphs coming up, looking at both ATP levels and the viable cell counts, which are both ways that we look at how a compound is affecting cells. When we first did testing with Recce 327 using our cytological profiling, which actually is a way of looking at cells on the individual level to see what's going on, the first thing we saw with 327 was that it was arresting basically all cell activity. So the cells were no longer dividing, they were no longer growing, but they were not lysing, which is something that you typically see with compounds that were affecting the cell wall or the cell envelope in some way. So this was a unique mode of action we had not seen before. That led us to start looking at things like cellular bioenergetics and whether that was being affected. And we found that Recce 327-1 affected cellular bioenergetics very rapidly and very strongly, and at the same time was also killing cells very rapidly. One note, we did all of the testing I'll be presenting is in 10% LB rather than pure LB, which is a very rich growth medium that we do a lot of our work in. We looked in 10% LB because there we were able to look at a range of species at the same relative concentration. So the concentration we'll be talking about is called the MIC, or minimum inhibitory concentration. And that's basically the concentration where you see essentially no growth when the cells are treated. And that's just a way so you can compare similar conditions across different bacterial species rather than looking at the pure concentration, which can be a little bit different between different species. So first up here is E. coli, which is probably most of you have heard of E. coli. It's a very common bacterium. It's in the human gut, and it also causes different kinds of diseases such as food poisoning very commonly. On the left here, you'll see a chart of the ATP levels in these cells under different conditions. So treated with no Recce 327, 1 times the minimum inhibitory concentration, 2 times the MIC, and then 5 times the MIC. And particularly with 2 and 5 times the MIC, you see a very rapid and substantial decrease in ATP levels. And then on the right, you'll see viable cells. So this is literally counting how many cells are still alive in the culture. And that graph is actually charted on a logarithmic scale. So it goes from about close to 10 billion cells per milliliter of culture to less than 200 cells per milliliter of culture. So Recce 327 is killing virtually all the cells in that culture within an hour when you treat with 5 times the minimum inhibitory concentration. So then we also did the same, basically identical testing in a different strain. This is Bacillus subtilis. This is a common soil bacterium, and it's a good model for a lot of gram-positive bacteria. And again, you see the rapid decrease in ATP levels when you treat with 327, and then a concomitant drop in viable cells. Again, we look in Pseudomonas aeruginosa. This is interesting. This is a wild type version of Pseudomonas aeruginosa. This is an extremely resistant pathogen. Very few drugs affect this pathogen. And we're seeing, again, very strong effects from 327. And then Acineta baumannii, another gram-negative bacterium. Klebsiella pneumoniae, again, you're seeing the same thing. Rapid decrease in ATP levels correlated with a rapid decrease in viable cells. And when I say rapid, I mean we're seeing a very significant effect virtually as quickly as we can test this. So within 10 minutes of treatment, you're seeing very strong decreases. Staphylococcus aureus. And this is a specific strain. There were some technical difficulties testing with Staph aureus, which is not at all uncommon with Staph aureus. It's a difficult strain to work with. So the techniques we were using for the other strains were not really working in Staph aureus. So we had to use a different one. But again, in this strain, we see a very rapid decrease in ATP levels, and that is also correlated with a very rapid decrease in viable cell counts. So this was all testing in just regular culture medium. We also tested 327 in what are called stationary phase or non-growing cells. And this is very significant because many antibiotics will not kill non-growing cells. Antibiotics such as the penicillins, they affect the cell wall, but it will only kill bacteria if the bacteria are actually growing. And when you have an infection, you very commonly have a population of the cells that are not growing. So if you're treating with an antibiotic that only kills growing cells, it will have virtually no effect on that on the cells in that infection that are not growing. Recce 327, again, kills non-growing cells, and it kills them very rapidly and irreversibly. So that's very interesting. And then similarly, Recce 327 will kill bacteria that are growing in a biofilm. And again, biofilms form particularly on implantable devices, such as catheters or joint implants, and can be very difficult to treat with antibiotics. We use several different common antibiotics as controls: tetracycline, which inhibits protein synthesis; ciprofloxacin, which is a DNA replication inhibitor; cephalexin, another cell wall inhibitor; and then colistin, which is membrane active. It basically just permeabilizes cells. None of these were very effective against biofilms, but 327 was extremely effective against biofilms. So again, another encouraging indication for this, this compound. So again, in conclusion, 327 is rapidly bactericidal, in largely, if not completely, irreversibly so. Reduces viable cell counts very rapidly. The effects are dose dependent, and it has very broad spectrum. It's bactericidal, and it's effective against both growing and non-growing cells and cells in a biofilm. So this is a very interesting compound, and we've been really enjoying our chance to study it in our partnership with Recce.
I'm Phil Sutton. I'm Vice President of Translational Sciences at Recce Pharmaceuticals Limited. I'm going to give an update on 3 projects that I worked on when I was group leader at Murdoch Children's Research Institute. Specifically, these related to bacterial sinusitis, Helicobacter pylori, and Mycobacterium abscessus. So bacterial sinusitis is a very prevalent health problem which affects almost 29 million people in the US each year. This is a slide produced by a Clearview, but basically sinusitis is a mild infection or inflammation of the tissues that line the sinuses. This infection causes symptoms such as facial pain, congestion, nasal obstruction, and fever. And this highly prevalent condition can be treated by intranasal antibiotics, which provides rationale for the development of a novel antibiotic agent. There's a range of bacteria that can cause this condition. They're shown here at increasing frequency. There's others, but the most prevalent infection that can cause bacterial sinusitis is Streptococcus pneumoniae. There's a subset of patients that develop severe complications, such as high persistent fever, inflammation, and altered mental state. And immunocompromised patients are at a higher risk for severe disease and can warrant immediate antibiotic therapy. So Clearview's interviews with key opinion leaders identified that R327 could have strong utility in poorly managed recurrent patients. Physicians were enthusiastic about the intranasal route of administration, which has been shown to work for R327. They found that the maintenance of activity with repeated use would be a benefit. And also, what was really important was R327's demonstration of its efficacy against a range of drug-resistant pathogens. So we've done several experiments in mouse models to show efficacy of R327 against Strep pneumoniae, the common strain that causes sinusitis. The first one shown here was performed by an independent CRO. In this study, nasal cavities of mice were infected with Strep pneumoniae. Then the mice were anesthetized while treated with R327 via both the intranasal and intravenous routes. And these treatments significantly reduced nasal infection by Strep pneumoniae. So on the graph, the blue is intranasal treatment and the purple is intravenous, and reduction is compared to the control in red here. And the reduction was similar to the positive control with azithromycin. In a second study, this was the one done at my institute at MCRI, we modified the protocol to improve it. The change that we did was we didn't anesthetize the mice. When you anesthetize mice, you increase the clearance of anything you put into the nose. So by not anesthetizing, you can increase the exposure of the bacteria to the compound that you're delivering. So in this study, mice were infected with Strep pneumoniae. They were treated nasally twice daily for 5 days with R327, and importantly, the mice were not anesthetized. When we did it this way, we got significantly reduced nasal infection of their Strep pneumoniae, and we managed to eradicate infection in 8 out of the 12 treated mice. And importantly, this was also superior to the positive control group that were treated with azithromycin. So the key findings so far is that R327 treatment by the nasal route can eradicate nasal strep pneumoniae infection, the most common bacterial cause of sinusitis in mice. Also, a very interesting observation, we have managed to successfully treat a patient who had a multi-drug resistant form of Pseudomonas aeruginosa sinusitis. So this patient was treated under the TGA Special Access Scheme Category A, and the patient reported a substantial reduction in infected discharge with no side effects after 3 days treatment with R327 via a topical nasal spray. So these data, which are sufficient and compelling, we believe, to potentially start towards a phase 1/2 clinical trial. Moving on to H. pylori. So H. pylori is a gram-negative bacterium. It infects during childhood and typically colonizes for life. An estimated half the world's population is infected, so it's an incredibly prevalent pathogen. The important feature of this infection is that it causes a chronic gastritis, that drives a number of pathologies. The key diseases caused by this are 2 types of cancer. There's a stomach adenocarcinoma, which is a major killer. There's a rarer cancer, gastric malt lymphoma, and the peptic ulcers, which comprise the gastric and duodenal ulcers. This infection can be treated with oral antibiotic cocktails, quite complicated treatments, which are typically a triple or quadruple therapy. And while the treatment can be effective in maybe 70–80% of people, antibiotic resistance is a large and increasing problem, and it's getting so bad that recently the WHO listed H. pylori amongst the top 10 bacteria where new antibiotics are most urgently needed. So we did some studies exploring first the efficacy of R435, an oral formulation against H. pylori strain in culture, and showed that R435 was very effective at killing H. pylori. We tested it against 7 strains. 4 of these were fresh clinical isolates and 3 of them were lab strains. And pretty much on average, R435 was as effective at killing H. pylori as 2 antibiotics, metronidazole and clarithromycin, which are frontline antibiotics used for treating H. pylori infection. Moving on to mouse studies, we treated H. pylori infected mice with R435, and we did get a significant effect. The infection was reduced, but it was only reduced by 1 or 2 logs. We did this on several occasions and got similar results. And this contrasted with treatment with positive control metronidazole, which did eradicate infection. So what a summary of these results tells us is that R435 is very good at killing H. pylori, but we don't think that the antibiotic is currently finding its way to the stomach with the formulation that we're working with. So we need to do more formulation work to improve the way it's delivered so we can get more of it to where the bacteria live so we can improve the killing. Mycobacterium abscessus is a member of the NTMs, or non-tuberculous mycobacteria. Basically, NTMs are mycobacteria that cause chronic lung disease that aren't the mycobacterium that causes tuberculosis. These can cause a range of symptoms, including chronic cough, weight loss, fatigue, fever, night sweats, shortness of breath. And importantly, NTM lung disease is an orphan indication with significant unmet need. There are a range of different NTMs, but we think the most important one to develop a new treatment for is M. abscessus. So the incidence of specific pathogens of the NTMs can vary by region, with M. abscessus most frequent in the West. And the reason for that is M. abscessus is a major pathogen in cystic fibrosis. And cystic fibrosis is the world's most common genetically inherited disease, which mostly afflicts Caucasians. Now, Clearview's interviews with key opinion leaders have perceived significant unmet need for novel agents that could be met by R327. In particular, starting at the bottom here on the left with increasing need, they believe that new treatments should be more efficacious and convenient. There should be therapeutic regimens which are safer and more tolerable, and also the novel therapies should have shorter treatment courses, because currently the treatment courses can be incredibly long, even up to 1 or 2 years to treat. So the current treatments that are given tend to be very toxic and very long. And if I can draw attention to the bottom right, there's a quote here: if a patient has M. abscessus, they're fortunate if they get any improvement, and there's sometimes potentially permanent damage. So with this in mind, we've been doing some research to investigate whether R327 could be used to treat M. abscessus infection. And first off, we did some culture experiments where we successfully demonstrated that R327 could kill M. abscessus in culture. And then we did some important work where we investigated whether R327 could kill M. abscessus while they infected macrophages. Because in the human lung, M. abscessus infect macrophages, which is a type of immune cell. So inside these cells, the bacteria are protected from immune attack. But not only immune attack, many antibiotics also cannot enter the cells and therefore don't reach M. abscessus. This is, in fact, is one of the reasons that a lot of antibiotics don't work against M. abscessus. They can't get to them to kill them. So we used human stem cell-derived macrophages. We used these cells because they are physiologically very similar to macrophages that are found in the human lung. These cells were infected with M. abscessus and then treated with R327. So 1 or 3 days after treatment, live M. abscessus inside the cells were quantified by colony-forming assay. And what we found was, if you look at day 1 on the top right, we got a very nice dose-response killing of M. abscessus inside the macrophages, which by 3 days had resulted in complete eradication in some of the groups. So R327 was extremely effective at killing intracellular M. abscessus. There was no toxic effect on the macrophages, and we saw a dose-dependent killing. And this, importantly, was also superior to clarithromycin-positive control. After that success, we've moved on to mouse experiments. So we've done a pilot study where mice were infected in the lungs with M. abscessus. They were treated with R327 nasally twice daily for 5 days. And then the levels of M. abscessus in the lungs were quantified colony-forming assay. This was a pilot study. We didn't optimize the dose, and we didn't optimize the infection. So we're very pleased with the result because no adverse events were observed in the mice while treating a lung infection. And what we found was the R327 treatment significantly reduced M. abscessus levels. And despite using unoptimized dose and delivery, the efficacy of R327 was only slightly inferior to that of the positive control. It's very, very similar effects. So to summarize that, M. abscessus has emerged as one of the most important lung pathogens in cystic fibrosis. It's a major driver of disease progression. The pathogens drive an inflammatory response, which accelerates disease in cystic fibrosis. If you can control the infection, you can slow down disease development. And it's thought that 20% of patients with cystic fibrosis are infected with M. abscessus. It's a contraindication for end-stage lung transplantation. Advanced patients with cystic fibrosis often need a lung transplant to stay alive. If they have M. abscessus infection, they're not allowed to get a lung transplant. Current treatments are ineffective and toxic. So a treatment that can eradicate M. abscessus that is not toxic and is quickly effective would be a major step forward for these patients. So R327, there was no toxicity observed against treated human macrophages or infected mice. We demonstrated very good activity against intracellular M. abscessus within human macrophages, with complete eradication of infection achieved. This demonstrated that R327 was able to penetrate the cells and kill these bacteria inside the macrophages. And we also achieved proof of concept, where we showed that killing of M. abscessus infection in mice was possible following nasal delivery. So the summary for the 3 projects. For bacterial sinusitis, we have some very compelling data, which points us to start a phase 1/2 clinical trial, hopefully in the first half of next year. For H. pylori, we've demonstrated very good killing of the bacteria, but further formulation work is required to improve delivery to the site of infection. And for M. abscessus, proof of concept has been very clearly achieved, and there's very compelling data to progress forward, hopefully towards a potential human study in the future. Thank you for your attention.
Yeah, hi everyone. I'm Guillaume Van Tagem, Managing Director at LifeSci Advisors, based in Switzerland. As a reference, LifeSci Advisors is an investor relation firm, and we have been working with Recce for now a good couple of years. Personally, as a background, I'm a pharmacist by training, and I spent 10 years on the sales side as a biotech analyst, mostly for UBS in Switzerland and London. And I used to cover actually antibiotic company, so I'm relatively familiar with the antibiotic space. One thing actually, as a preamble, actually, I would like to discuss is the interest or the lack of interest of investors for antibiotics in general, I mean, historically, which is mostly due to the fact that although the development of antibiotics has a relatively high success rate, from a commercial point of view, the antibiotic space was hit by the fact that new antibiotics, because of the fear of resistance, tend to be kept last, right? So the peak sales, I mean, the sales trajectory of antibiotics tend to be relatively slow. Where I think actually Recce differentiates itself from the crowd is very much about the concept of almost no, I mean, conceptually, no likelihood of development of resistance. That's the difference between an antibiotic and an anti-infective, which Recce's compound is. And so therefore, I think that Recce, from a commercial point of view, will have actually a very differentiated commercial profile compared to the antibiotics as we know them. So I was asked actually to discuss the urinary tract infection market, okay, which sounds relatively silly, but is a very, very large market. Okay, it's one of the most common infectious disease. And while in most cases, actually, it's considered as a relatively mild infection, actually can go bad relatively fast, not only because of the lack of resistance, but as well the fact that women, mostly women, actually get reoccurrence over and over and over again. And sadly, for quite a few, actually ends up with kidney issues, kidney failures, and so on. As you may be aware, one of the most common infection is, this infection is caused mostly by a bacteria which is well known called Escherichia coli, that accounts for roughly 60 to 2/3 of the UTI infections. And that's basically a normal contamination from the gastrointestinal tract up to the genitalia and the urinary tract. While actually Escherichia coli used to be very sensitive to most of the classical antibiotics, the beta-lactamase, the beta-lactam, the tetracycline, and so on. Over the last 20 to 30 years, a significant increase in resistance has occurred. If you look at ampicillin, 86% now are resistant. 86% of Escherichia coli are resistant to ampicillin. Amoxicillin, 76%. I mean, it's a relatively old drug, but still, by the way, developed by GSK. Tetracycline, and so on. I mean, you have the list here. Now, globally, actually, that's a very, very large issue that affect almost basically more than 400 million women in 2019 in the world, right? And from a cost point of view, while actually the cost of the antibiotics that I mentioned are mostly all genericized, if not all, still, so low price, it still costs $6 billion in direct healthcare expenditure, mostly related to the antibiotic treatments. What is more and more worrying is that the resistance level is relatively high, but it keeps on getting worse. One of the issues as well of women is that because UTI are so common and reoccurring, okay, they tend to use always the same antibiotics, creating actually more exposure to the bacteria, to the same antibiotics, therefore leading to more and more resistance. Okay, so and we end up actually with one key issue, which is 1 in 3 uncomplicated UTIs are resistant to Bactrim, which is considered to be one of the standard of care in UTI, and 1 in 5 now are resistant to 5 other common antibiotics. And so that's a very, very big issue, because if you look at the list of antibiotics, we are starting to run out of options for these women or for these patients. So if you look at the market of the UTI, so we know that the UTI cases are rising, okay, roughly 4-5% at the CAGR a year. That has to do with the population growth, more access to healthcare as well, more awareness, so more detection. And so actually there's an increase cost, right? I mean, the market size was roughly $9 billion in 2020, and we do expect this market to go to 12 billion by 2027. If we look at specifically Australia, for our Australian colleague, it cost UTI alone almost $1 billion Australian dollars, and that was actually I think 2 years ago. There has been a number of players in the field, though, okay, of antibiotics, most notably actually GSK. I mean, you have the list here. So there's a big interest in UTI, yet the degree of clinical development in UTI has been relatively limited. All the commercial issue I was mentioning before. Now, there's one thing actually I would like to bring to your attention is the recent deals that was announced, right? That came as a big surprise of GSK entering an exclusive worldwide collaboration with Spero Therapeutics, who had actually developed and submitted for regulatory approval an antibiotics against complicated UTIs, right? And who paid actually $66 million upfront and additional milestone and tier royalties. And I'll come back to that later. What is interesting actually is that Spero had generated a positive phase 3 clinical trials. I mean, technically, they demonstrated that they were statistically non-inferior, which is a standard FDA requirement, so non-inferior to the standard of care. And despite this positive trial, they submitted and received what we call a complete response letter from the FDA, questioning the quality of the data. However, following actually what they call an encouraging US FDA regulatory feedback regarding the next step of clinical development, okay, GSK thought it was doable to rerun a phase 3, right, and to generate sufficient data to get to regulatory approval. One thing is that, this drug from Spero will be the first oral carbapenem antibiotic to treat this indication, which is complicated urinary tract infection, okay? And that includes actually complications such as pyelonephritis, okay, so affecting the kidney. So if we go back actually to the details of the deal, and I think it's particularly interesting and gives you actually a bit of balance as to the sentiment of the market on the anti-infective space. So before the deal, right, was announced and following the complete response letter from the FDA, so basically the FDA refusing to approve Spero's treatments, the market cap of the company had fallen to $28 million, right? So it was not in great shape, but the deal actually with the $66 million upfront payment from GSK, and actually it's even more because it's $66 million cash plus $9 million equity investment, the market cap only increased to $73 million. So what's interesting is that indeed Spero will need to finance the phase 3, the new phase 3, but with the cash injected by GSK, they have the mean to run this phase 3. It could be actually a phase 3 that could be run actually relatively fast. Okay, I mean, they know the market, and it's as I said, it's a common infection, right? But so what's interesting is that the market cap only increased by 60% of what GSK had injected in cash, leaving alone any increase of probability of success, any impact of having GSK on board. GSK actually is very familiar with commercializing antibiotics, despite the fact that there's a lot of upcoming milestones and royalties and so on. So the market is still actually relatively dubious about the likelihood of having a drug, an antibiotics with a proper market. Okay, but what was interesting in this press release as well, it's really rare for companies, especially large cap companies, to allow to disclose actually much details about the upfront, about the milestone, and about the royalties on sales. Here on the right of the screen, actually, I've listed, it's exactly a copy paste of the press release, where they list actually step by step the financials that Spero is eligible to receive. Okay, I mean, one thing which is interesting is as soon as the phase 3 will be delivered, right? One will argue in 2.5 years, probably. That's my assumption. Spero is eligible to receive $150 million, right? If we look statistically at the likelihood of having actually a positive phase 3, especially that they already had a positive phase 3 based on non-inferiority, that is relatively high probability of success, yet GSK feels comfortable to chop in actually this kind of amount. And the same actually once the drug is approved and the first commercial sales occur, then there will be an additional $150 million. Another thing which is interesting is that on top of the low single digits to low double digits royalties on sales, which one will argue is relatively low for a phase 3 compound, but still, given the history, actually can be justified. GSK is happy as well to chop in up to $225 million. Okay, so I don't think actually that based on that, GSK think that it's going to be a mega blockbuster, 2-3 billion, right? But still, only in that indication, one would argue that they still expect to be between $500 and $1 billion, which is actually something that analysts don't really have. So to go back to it is that the market actually for anti-infectives is very important. This one actually is a carbapenem actually conceptually could develop resistance, unlike the Recce compound. And so despite that, actually, GSK, who is famously known for being good in developing antibiotics, is quite bullish on the market and is ready actually to put some capital at risk with measured risk and to move forward. So I think that the reason why I wanted to bring this GSK Spiro deal here is that I think that the sentiment behind antibiotics is starting to change. The interest from pharma is increasing. And I think that Recce is very well positioned, not only from a clinical development point of view, but as well from a pure technical point of view with a low risk of resistant development to attract a lot of interest from commercial players in Europe and in the US. Thank you very much.
Well, thank you all. My name is Michelle Delizia, Executive Director and Chief Scientific Officer from Recce Pharmaceuticals. To summarise then, the scientific strategy of Recce is to build upon the very many opportunities that its breakthrough anti-infective technology provides across a range of infectious indications and unmet medical needs. This is seen in a strong pipeline of current preclinical and clinical studies, which include not only antibacterial work, but important studies in antiviral programs and across a range of administrative routes. We're going from serious life-threatening bacterial infections for our lead candidate, Recce 327, topical applications, investigations into serious unmet medical needs for infections relating to Helicobacter pylori, Bacterial Sinusitis, and a very tough organism, Mycobacterium abscessus. A compelling and true-to-life example of one of the indications for Recce 327 is in sepsis. And in this awful patient case study, the patient woke up with a raging UTI that had progressed to sepsis, and she was critically unwell. Typically, patients in this scenario are critically unwell. There's bacteria in the bloodstream throughout the body, and the identity of the bacterium, or often multiple bacteria, is not known, it's not identified, and nor is an appropriate antibiotic known to effectively treat. Now, Recce 327 is uniquely placed to uniquely treat and save lives in this scenario because it has this unique and universal mechanism of action. It can be administered straight into the bloodstream with confidence, with physicians knowing that it will effectively and quickly treat any bacteria that's causing infection, that's causing a threat to life, even resistant superbugs. And in addition to urinary tract infection, which accounts for about 25% of cases leading to sepsis, there are many other pre-septic conditions that are indicated. For example, pneumonia, surgery and wounds often can lead to sepsis, burns, abdominal infections, and meningitis. In recognition of Recce's brand new class of anti-infective technology, global patents have been awarded. More than 40 have been issued, offering protection for the technology in significant global pharmaceutical markets. So the way forward to tackling the global health threat of antimicrobial resistance of superbugs, of unmet medical needs, is to invest in a truly new class of anti-infective weaponry. Recce's anti-infective technology delivers, for the first time in more than some 30 years, a unique and universal mechanism of action to quickly and effectively treat bacteria across a broad spectrum, including superbugs, in a great range of therapeutic indications. Undoubtedly, there are significant market and commercial opportunities, but perhaps more so, there is the opportunity to contribute for the benefit of world human health. Thank you for joining.
Thanks, Michelle. That concludes today's presentation. These slides have been made available on the ASX and will be uploaded to the company's website. We would like to thank you for your interest in our anti-infective company and look forward to bringing to you expansions and advances in our technology platform over the time ahead.